What I actually want to do today is describe to you how we use pluripotent stem cell-based assays for early-stage drug discovery, and referring back to the title of our symposium, really look into the future, what iPS technology or pluripotent stem cell technology has to offer in terms of drug discovery, and how we see it, how we apply it already today together with our partners. And I just try to reiterate what drug discovery is about and what people have done in the last decades, and basically just reminding you that it takes very long to get a new drug to the market, so a new molecular entity, NME. It's very, very expensive, and a lot of money is actually needed in later clinical development. But also even in the early stages, there is a substantial amount of money needed to do this. There's a substantial amount of clever projects to go on, and also some time is required here. And we still see a need of good academic ideas, of good disease models being actually translated to finding new medicines and applying these to provide better medicines in the market. So what we do in Hamburg, in our Fraunhofer site there, is basically this model that we work together very closely with academic partners, that we try to develop jointly starting points for drug discovery, and that we take their biological assay, their biological asset rather, their idea, their concept, and try to make a very robust assay which is amenable to high throughput screening. We have the automation ready to do that. We have a large collection of chemical compounds. I will describe to you in a second how we do that. And we also have a screening facility, plus the personnel that is years and years of experience in pharma and also in academia in chemical biology. And our ending point is basically a validated hit. So if you have a hit, it basically means nothing. So you have to validate your hit in orthogonal assays until you're very confident, at least, that there's maybe good reason to test this compound in vivo efficacy assay. So we usually then partner with other financially stronger partners to move this forward into lead identification and also to nomination of clinical candidates. So it's just a snapshot of how it's set up. The screening technology that we have in-house is basically a customized PerkinElmer system, fully automated, and we have both chemical and cell-based readouts. But we also have the technology to handling very large quantities of small molecules. Over the last 4 or 5 years, we also moved into cell-based assay for toxicity assessment, and we moved away from traditional hanging drop-based methods to more automated technologies using in spheroplates plates and also a TCAN system, where we can now start to manipulate 3D aggregates and try to expose them to small molecules, in particular for toxicity testing, but we can also hopefully in the future go beyond this. So compounds, that's always a big topic, and we've heard about repurposing strategies, but I just wanted to point out that we have a large collection of both lead-like synthetic compounds, natural products, but also known drugs for repurposing. And I don't want to go into all the different details here, just mentioning briefly that we are also an EU Open Screen partner site, which is a European infrastructure initiative where we also provide or will provide in the future a customized library also for academic partners to screen these. And I wanted to particularly highlight this new effort from the Broad Institute, which is the most sophisticated repurposing collection to date that has been published just earlier this year, and it's called the Drug Repurposing Hub. We'll be the first partner in Europe basically implementing this broad library. And the strength of this library really is that they went to the length of identifying all the compounds that are A, on the market, B, that have failed clinical development, but not for reasons of toxicity, but for reasons of safety. Because of reasons of efficacy, but they're safe, and also compounds that have been on the market, but that have been withdrawn from the market. So it's a very valuable collection in our eyes, and the secret really behind that is, Christine was also alluding to that actually the quality of the compound also does matter, and these guys put a lot of effort into trying to understand what is the best vendor, who provides the best quality, and then also, what is this compound actually hitting, which target, which disease class, and also a lot of metadata is annotated in this collection. So what do you do with all that data? Of course, you need sophisticated data management tools, and we have a high-content data management system which basically links all this information between the biological, the chemical, and the imaging data that we retrieve. And you can then move ahead and can do all types of analysis, and can go back to your image, can go back to your dose response, and really zoom into the properties of that compound. And we are set out to do so in Hamburg. So I don't want to embarrass you, but let's look into this model of how future medicines can look like. And we are, of course, mainly interested in this latter part, so deriving iPS-based products, generating disease-affected cell types or their isogenic controls, as we heard, and then mainly developing assays, running high-throughput screens, validating the hits in dose response, and providing good starting points for drug discovery. This is what we do. So let's look into some data that we generated jointly with our partners. And I'm quite happy that I don't have to touch the cardiovascular field for now, but I'm more focused on the neurodegenerative part of iPS technology, or where iPS technology can be applied. And I want to talk about, in my first example, I want to talk about multiple sclerosis, which is a chronic, basically a neurodegenerative disease which is caused by a chronic CNS inflammation. So you have immune cells that behave weird in the central nervous system, and they, in the end, lead to a loss of oligodendrocyte, loss of trophic support, and eventually to axons that die, and to neuronal damage over time, over decades in most cases. And the cascade of molecular events leading to this damage is more or less clearly defined, and at least this is our version of the reality, that this chronic CNS inflammation creates a milieu of, on the one hand, reactive oxygen species, of hypoxia, cytokine overload, pro-inflammatory cytokines, but especially glutamate stress. So you have a high concentration of glutamate that leads to, in particular, some calcium influx into the cells, ion channel redistribution, energy deficiency, and then an overall imbalance of the ion situation, basically. And in the end, oncotic cell swelling and neuroaxonal damage, and death by apoptosis or necrosis. And this can be clinically, and I guess you all have seen these deep stuff images. This is basically MRI images, which indicate then if you have a dye, that certain regions of the brain are just not myelinated, and the neuronal substance is lost in these areas. So in the last decade or so, a lot of these underlying molecules have been identified that are causally linked to neurodegeneration. A lot of ion channels are actually now being thought of to be causal for the development and progression of the disease. And we have studied jointly with partners 2 ion channels, ASIC1 and TRPM4. ASIC1, I think Andreas was also involved in that project in the beginning, and jointly with Evotec, and then later on with Merck's Rono, we identified compounds that actually did show in vivo efficacy, but in the end the program was stopped because Merck's Rono just discontinued their efforts in neurodegeneration. So you can, even though you have a very clever approach, doesn't mean you get a drug to the market, right? So let's focus now on TRPM4 and how we think it's contributing to neuronal decay. So I told you that there's a high load of glutamate in the inflamed central nervous system. This is triggering NMDA receptors, leading to calcium influx. This activates ion channels of the TRPM, or the TRP ion channels, in particular TRPM4, which then shuttles in sodium and other monovalent ions, and is leading in the end to cell death. There is a dirty drug that was supposed to work in the system, which is glibenclamide. Glibenclamide was the most prescribed anti-diabetic drug in 2003, so it's a blockbuster drug on the market. It's not targeting directly TRPM4, as people have realized nowadays, but it's targeting the sulfonylurea receptor, SO1, and thereby mediating its function. But it's not as efficacious in vivo as you would hope. So basically, this is a project that started in 2009 with our partners, and they saw when they knocked out this TRPM4 ion channels in the model of multiple sclerosis, the experimental autoimmune encephalomyelitis, that these animals were largely protected from the progression of the disease of multiple sclerosis. And you see that also here, that if you have now cultures of neurons from these, so this is all a mouse, right? From these neurons, that if you now supply them with glutamate stress, and then the TRPM4 knockout neurons are largely protected. And also, you only see this oncotic swelling that I mentioned earlier. You see this only in the neurons that are wild type, but not in the TRPM4 neurons. And also the disease model now, again, in the mice, when you treat them with glibenclamide, with this dirty drug that I mentioned, these animals are largely protected, just as if you would just knock out TRPM4, indicating that this could be a way to prevent the disease onset and progression in multiple sclerosis by modifying the function of TRPM4, in particular pharmacological inhibition of TRPM4. So this is what we set out to do in 2013, jointly with Evotec. So we were screening approximately 250,000 compounds in a high throughput screening campaign. Again, using hex cells. It's just the way how you still do things. You cannot screen 250,000 compounds using iPS cells just now. It's just not feasible. And we used a flipper-based strategy by the ion-sensitive dye, monitoring basically the fluorophore uptake of these cells. And we got a couple of hits, but a hit, as I told you, a hit means nothing. We confirmed these hits in a high-throughput electrophysiological Q-patch assay, and we came up basically now with 5 lead series that show potency in both these assays that are specific for TRPM4 and that do not hit TRPM5, which is the closest relative here. And we specifically in this program, we implemented iPS-based methods, iPS-based models to really prioritize these compounds for their efficacy in humans, at least in an early stage of the program. So we don't want to just see a rescue effect of these compounds in primary mouse cells, but we want to see a rescue effect in human iPS-derived neurons before we go into an in vivo model of multiple sclerosis. And this is basically how these classical efficacy and ADME sheets look like in pharma, right? So you have a bunch of assays that you have to go through, and you just determine the corresponding values, and based on these properties of these compounds, you progress a compound, or you basically kill it off. So, but what we really want to do in this program is implement iPS-based disease models to show the protective effect of the compound in such a setting, and also to enable compound decision-making and progression of compounds to in vivo models. So that's what we did. We compared different differentiation protocols, and as we learned, it's actually quite nice to work with neuronal precursor cells because they can still differentiate, and we can grow relatively large quantities of these cells. And we compared different protocols to doing that, and we found that a protocol published in 2016 was particularly suited to derive cortical neurons for our purpose, so they look okay in terms of their marker expression. I cannot show you functional data at this point, but what I can show you is that these cells actually do express TRPM4 channels, which was not reported previously in the literature. And in addition, they also express NMDA and AMPA receptors, at least on an RNA basis. So we're quite confident that we can use this model to progress. Keep in mind that TRPM4 is also highly expressed in cardiomyocytes. So that might be a potential liability for the compounds. There is a gain of function mutation of TRPM4 associated with cardiac phenotypes, but this is something we just have to find out, right? So it's not super clean. It's not an ion channel. It's solely expressed in the central nervous system, but it's a rational target. And let me guide you through this busy slide now. To really test the effect of the compounds, we went then on to basically generate cortical neurons in 96 well plates, and we used this so-called real-time glow substrate, which is provided by Promega, which is a non-lysing substrate to assess the viability of a cell, so that the dye enters the cell. If it's reduced, it produces light. If the cell is dead, it doesn't produce light. And this is a typical rescue experiment that we can then do in a kinetic type of way. So we put a little stress, a little glutamate stress to the cells, and you see that if you just have glutamate, 50 micromolar glutamate in your culture, 30% of the cells die. If you use APV, which is a very potent blocker of NMDA receptors, then you basically rescue your culture to the full extent. And if you now have our compounds that are inhibitors of TRPM4, you have somewhat of an intermediate effect. And this is actually what we're looking for, to have the best rescue possible. And this is just summarizing up a couple of different experiments with the individual compounds. And you see here these gray bars is just an average of the rescue that you can achieve. So that actually gave us quite a confidence that our screening strategy was more or less successful until this point. But what we also found nice is that the IC50 in the primary assay, so in the primary flipper assay, corresponded nicely with the rescue effect of the compound in the iPS-based model. And of course, based on these findings, we will now prioritize compounds and move them on into in vivo studies. The second part of the cell-based, so this was more the biochemical assay in a way of showing rescue simply based on viability, but there are also more subtle changes in the cell-based models that you can tease out. And what we do a lot is basically high throughput microscopy, high content imaging, and there are tools by PerkinElmer, but also from other providers that are actually nowadays very easy and make it like a drag and drop functionality to, on the one hand, identify individual neurons, but also quantify the length of their neurites, their branching points, the neuronal network, as it were. And you can do that quite easily nowadays, as I said, pointing out individual neurons and their phenotypic properties. And if you now, this is just an example now, using a very old known TRK tyrosine kinase inhibitor, that you actually, by titrating this in, you actually lose the total neurite length. So you can also use high content parameters to tease out the functionality of these compounds in terms of rescue of neuronal loss. And this is what we plan to do. But what you can also do this in vivo nowadays. And there's a very nice instrument by Essen Instruments. It's called the InQSight, which was bought by Sartorius, and you can do then can basically track neurons over longer periods of time, so over days, and you see the similar rescue experiment that I showed you previously. So full glutamate exposure, the cells basically die. Rescue now with APV, this is the blue line here, two different concepts, DMSO and APV rescue, and you see start to see somewhat of an intermediate phenotype of these compounds that show this rescue effect. And if you now look into this top right corner, there are also different basically live cell dyes that people have used, and you can then also track the dying cells basically over time. This is just now a Cas-based substrate that is accumulating in the nucleus, but it gives you quite a nice kinetic in vivo assessment of your rescue effect, and we are trying to implement those models as well in the future. So let me switch gears and talk about the second part that I wanted to address. We heard about endothelial cells already. We heard about barriers. And I also, if you paid close attention, I showed you that these PAMPA assays, so parallel artificial membrane permeability assays, are very easy and very standardized instrument in early stage drug discovery. And compounds are prioritized based on these findings, because however clever your strategy is to target the central nervous system, you have to overcome the blood-brain barrier. And you have to reach your target at decent concentration in order to prevent the effect, the disease effect, if you wish, in the central nervous system. So what a PAMPA assay is, basically you have a acceptor well, you have a donor well where you put your compound in, you have an artificial phospholipid membrane, a bilayer, and substances are basically diffusing through these barriers. And you can then prioritize compounds based on the transcellular flux. And you have, of course, substances that traffic very slowly through the barrier, moderately or highly. But the reality looks quite different, as we heard also. Now that the neurovascular unit is very well organized, is very tight, consists of specialized endothelial cells, parasites, but also neurons, astrocytes that are kind of touching the system, make it very tight, make it very restricted compartment. So what you rather want to do is do something like this here and try to put this in an in vitro model. So again, summarizing the arguments for maybe using iPS cells at the blood-brain barrier for drug development is that non-predictive models actually also cause the high attrition rates in pharmaceutical development because you simply cannot get your drug across the blood-brain barrier. The brain endothelium represents not only a physical barrier but also a metabolic barrier because it contains a polarized structure. It's expressing a lot of transporters, for example. And people have reported barriers of up to 2,000 ohm per square centimeter, so basically measuring their transendothelial electrical resistance. But what you also want to assess, you don't want to only measure the transcellular uptake, so to say, to the other compartment, but you want to also measure the paracellular diffusion of these cells. So it could look something like this, that you have a complex cell-based model, you have an apical, you have a basolateral chamber, and you have a complex model at the interface. And this is basically what we set out to do this year, especially with partners in Würzburg, Antje Appelt-Menzelt, Marco Metzger, that we, on the one hand, produce brain microvascular endothelial cells in the apical compartment, but also combining that with the other cell types, iPS-derived, and then, on the one hand, validate the system by measuring this TIR value, by providing or by applying labeled tracers or approved drugs that we then try to pick up at the basolateral side. And then obviously this is also great for disease modeling, right? So that we just take a control, an isogenic control of a patient, and of course we have some genetic variants or some genetic cases of Alzheimer's disease where we really want to see what's going on at this blood-brain barrier, which transporters are misexpressed. Is what is the disease situation looking like, at least in this in vitro model? And we have people previously have described this barrier only with the IME-94 clone, but we also recapitulated this phenotype with our in-house line. Again, our in-house reference line, we had this discussion yesterday, was a reference line. We have an internal reference line, obviously, as everyone else. And with these, we try to miniaturize the system so that we can look in parallel at a lot of different types of compounds. Also in dose response, very important, because otherwise you have to produce tons and tons of plates. But until now, actually it's a problem of material because we cannot get hold of decent 96 well plates that have this insert configuration. That is where you get a tight endothelial barrier. But we're quite confident that with this Griner, 24 well plates at least, we get very nice transendothelial electrical resistance values, which are in the range of what Antje Appelt-Menzelt has reported earlier this year in Stanford reports, which is more or less getting closer to the situation in vivo, the status from the early 90s where people have reported values of larger than 1,500 ohm square centimeters. So that's where we're heading. So on the one hand, building more sophisticated in vitro systems, in vitro barriers for blood-brain-based drug discovery, but also for disease modeling, and then also a combination of both. Yeah, this is basically just also showing you that the transport study is also important, that you also have to determine permeability coefficients that then also report on the integrity of the system, and this is also quite well in accordance with the published data. And of course, then as an outlook, we want to receive blinded compounds from a pharma partner, similar to what we heard in the cardiovascular field, and want to then assess together with AbbVie, are these in our model, is our model superior in a way to predict the blood-brain barrier penetrance of these compounds? So lastly, I wanted to quickly touch on a topic that Kevin mentioned yesterday. Now completely switching gears, but just to tell you what so that we basically now try to apply the lessons learned from EBiSC, and I think it's been a great interaction, great training sessions, great teamwork, and we try to apply this in a very different setting with a CRO, also based in Hamburg, Evotech, and which basically approached us and wanted to have also cell lines for their drug discovery efforts, and wanted to have a rigorously controlled cellular product, and that's basically what we're doing for them. So not only the flow cytometry analysis, the standards that we identified in the consortium, but also the high content image-based assessment of pluripotency, for example, of marker expression, if you differentiated these cells. But in particular, we're also performing pluritest analysis for Evotech. And just quickly mentioning that pluritest is a widely accepted tool. Maybe if I switch to the next slide, widely accepted tool now since 2011. It's used by a lot of people. A lot of data sets have been deposited in this tool, and it's basically again a quality control tool, which is telling you whether a cell is pluripotent or whether it's not pluripotent. And a lot of partners also are using this locally, and it's also accepted QC tool in key journals. If I can quote you on the ISKI initiative 2015, that this is actually fine when you also assess your cells with pluritest. You don't have to go into in vivo models. So we have implemented in this consortium, we have previously the pluritest was based on HT12 Illumina arrays, but we've now extended this platform, and I just wanted to mention this, to EFI, the new version of EFI chips, but also to RNA-seq-based methodology, and we have applied this also this year, and I hope that we will also provide an update paper basically on pluritest in the near future. Keep in mind, and we touched upon this with Ola's data yesterday, that pluritest can do certain things, but it cannot do everything, right? So pluritest is a tool for unbiased assessment of the transcriptional pluripotency state. It does not give you predictive information on the differentiation potential of a cell. And for example, reactivated transgene expression cannot be picked up by pluritest. This has been shown also recently by Christian Freund and others in Leiden. And what these authors suggest is basically now a combination of pluritest and scorecard for the assessment of your cellular product. At least that's their perspective. That I just wanted to highlight this paper that I find quite recent. So let's sum up. So we provide state-of-the-art, high-throughput screening and high-content screening facilities, which are usually typically found in pharma, but that we can also jointly with you use in your projects or for your academic ideas to move them forward to translation. We are open to everyone. We just have to find money to do things together. We have access to large compounds collections, including in the near future the broad repurposing collection. We try to integrate phenotypic screens and functional assays based on pluripotent stem cells into our drug discovery programs. And I showed you the example of TRPM4 ion channel, and we try to extend this to other areas, for example, the blood-brain barrier. And of course, we try to take the lessons learned from the last 3-4 years in EBiSC and apply them to new projects with a CRO in Hamburg. And with this, I would like to finish. Thank a lot of people that have been involved in this project. And I wanted to especially thank the EBiSC consortium, especially Glyn again for the training sessions that he provided, because I think it was a forum not only to learn about how to work with iPS cells, but also to meet people, to interact, and that a lot of joint initiatives have spun out of these training sessions. I think it was great work from you and your team. And with that, I would like to take your questions. Thank you.
Thanks very much, Ole. That was really very, very good. Questions?
Yes, this artificial blood-brain barrier. So how many drugs have been tested for permeability on this, and is that data freely available?
So basically, Pampa is just one system that's been used in pharma. No? So of course, you have other assays where you try to assess, what role P-glycoprotein plays for export of substance and other. This is just a routine assay that's been applied. And in terms of data, I mean, as I showed you now on this scorecard for compounds, it's basically run in every ADME program that you can think of, because it's an artificial bilayer. It's quite easy to do, quite nicely standardizable. There must be millions of data points using the pumper assay.
But do those live within companies, or is that available to somebody who's interested in a particular compound?
So in our consortium with the blood-brain barrier, we also have small, medium-sized enterprise partners that provide these services. Pharmaceuticals, for example, in Western Germany, they do that. It's a routine technology that basically a CRO can provide. Yeah. So it's a very common assay. It's just that iPS cells could potentially integrate all these different transcellular, paracellular, and transporter-mediated process at the blood-brain barrier, and it's not only highlighting just one aspect of how the compound can get across the barrier, but maybe integrating all these different pathways. That's just the idea.
Thank you very much.
Thanks.
Well, if there are no further questions, thanks very much.